the human body comprehensively at the single cell level. The HIVE has a structure that's made up of 4 components. The 4 components are the collaboration aspects, the infrastructure aspects for handling data, mapping aspects for getting the single cell data represented correctly within the right tissue and organ and system representation, and a tools component, which is everything that gets the data out to the community. The objectives of the HIVE will include things like manage the data generated by the consortium, coordinate internal and external consortium activities, develop tools for visualization, searching, and modeling data, and build the atlas of the tissue maps. So this slide covers at a very high level what the role of each of the components are. So in the collaboration component, the primary responsibility is to coordinate and promote collaboration within the HuBMAP consortium and the HuBMAP consortium with the wider research community. In the infrastructure component, the primary focus is to build and manage the data platforms and support the internal and external facing informatics tools that will be made available by the consortium to both the larger community and within the HuBMAP itself. In the mapping component, you'll work on integrating the tissue mapping center data generated that Zarina described and earlier is another component of the HuBMAP initiative, and with other existing data that's available outside of the HuBMAP program, and to build spatial maps of biomolecular distribution for the entire human body. And the tools component, you have to build and manage a suite of tools, analysis and visualization tools for the HuBMAP data. An important note is that each application may propose to apply to one of these components, or you can apply to more than one, and if you do, then you have to generate a coordinated set of separate applications. And the details of this are there in the FOA. More details are also there in the appendix. To be responsive to each one of the components, in the collaboration component, you have to propose inward and outward activities that will support collaboration, communication, and accountability. And in the infrastructure component, you have to propose practical, scalable, and flexible solutions that integrate imaging and genomics data and keep a cloud-first attitude towards building such an infrastructural component for the HIVE. In the mapping components, you have to propose integrating existing data with the newly generated HuBMAP data through the tissue mapping centers, and to put these data together into a multi-scale, body-wide spatial maps of multiplex biomolecular data. And the tools component, you'll have to propose a suite of search, analysis, and visualization tools for accessing data with end users in mind. Some more key points to keep in mind. As has been mentioned before, this is not a grant or a cooperative agreement. It's something called other transactions, which we'll get into later in this presentation. So it'll be actively managed and with much more input from NIH than other ways of making awards. It's important that the 4 components work together in a seamless manner and be prepared for a lot of intense interactions. So you have to think about, if you are proposing more than one component, how you propose to work with other components, even if those components are not part of your application. Flexibility is going to be key to the success of HIVE. As you may well realize, the data and the technologies that will be used to generate the data will evolve, as will the underlying data science and informatics tools and hardware and software and ways in which software engineering is done. And so therefore, you need to be prepared to work with the NIH staff to adjust, to add, and to delete activities and partners as the program evolves. And it's important to think about what practical deliverables you would propose in your application. So you need to think about what endpoints you will demonstrate, how you will use existing off-the-shelf tools. Think about how to modularize solutions that you propose and how they can be integrated into workflows and other harmonization activities that will be required. And with that, I will let Richard take over and describe a few words about other transactions.
Thanks, Ajay. To answer one of the questions we've had so far, all these slides will be made available after this webinar. We'll post them on the website, which is down at the bottom right, commonfund.nih.gov/HuBMAP. We'll also make a recording of this webinar available later in the week. So we want to say a little bit more about other transactions, because these are, as it says on the slides, not contracts, not grants or cooperative agreements. These are another type of award mechanism. And it's important that you be aware of some of the differences for what these awards are in comparison to some of the mechanisms you may be familiar with. So what are other transactions? So the reason why we're using other transactions for this particular funding opportunity is that it allows us the nimble addition or subtraction of expertise, tools, or technologies, as well as bringing in additional partners or expertise that we need as the program evolves. So the mechanism lets us be able to do that in a much more flexible way than we would be able to do, for example, as part of a grant or a cooperative agreement. It also gives NIH much more input into the process, and in terms of the details of who the major players are, you can look in the RFA, and it outlines the number of different roles and responsibilities of both NIH staff as well as awardees, and how we see that interaction evolving. A second reason for using other transactions is it facilitates engagement of non-traditional partners. So if you look at the details of the eligibility of who's eligible to apply for these awards, you'll see that it's a much wider swath of potential partners. For example, other transactions allow for individuals to apply for an award as opposed to institutions. So it's really that flexibility of engaging non-traditional partners as well as the nimble management of it that were the reasons for using other transactions for this particular funding opportunity. So what are other transactions? So as I mentioned, they're not grants, cooperative agreements, or contracts. So that means that we don't follow the normal NIH grants policy or the FAR. We have to establish a policy guide for the use of these other transactions, and we've included a link here. If you go to the HuBMAP website, you can see this policy guide. There are a number of general public policy requirements that all the other transaction announcements that NIH follows, so you can also see the list of those public policies listed on the website. And we have a separate set of frequently asked questions related to other transactions, and we certainly welcome your questions during this webinar or in follow-up by email. But I think it's important to recognize that these are not managed in the way that grants or contracts would be managed, and that you need to take that into account both in terms of how you're thinking about your application as well as talking with your organization representatives and thinking through what this means for them as well. So some of the key differences between other transactions versus other kinds of announcements: there's more flexibility in how we solicit applications and the submission format, and we'll talk a little bit more about that next. It also encourages non-traditional partners to submit applications. And so we try to make the announcements as flexible as possible and really targeted towards what we're specifically looking for. How these applications then are reviewed, the process is called objective review to distinguish it from the normal peer review process. It allows us to bring in a more diverse set of reviewers and to have interactive discussion as part of the review process. And again, we'll go into this in more detail. And it allows us more flexibility in deciding how to fund awards. So in terms of award management, it's a much more dynamic process in terms of how awards are managed. They can be expanded, modified. NIH can request partnerships, or partnerships can be proposed by awardees. And obviously, again, awards can be discontinued based on programmatic needs or NIH interests or by mutual agreement. And again, all of these, it's part of the reason for using other transaction is the rapidly evolving landscape of this particular field, the changing availability of funds, as well as the evolving programmatic needs based on the other awards that we make as part of the program. So I'm going to turn it over to Jessica, who's going to talk in a little bit more detail about the review process.
Thanks, Richard. My name is Jessica Smith, and I'm the OT SRO for the HIVE SOA. I work in the Office of the Director, and today I'm just going to briefly walk you through the process for LOIs, applications, and review. So this is just an overview of the process on this slide. Just to emphasize, the first step is discussion. Applicants are strongly encouraged to discuss potential projects with both Ajay and Zarina and Richard. The next step is letters of intent, which are due on February 28th. Those will be evaluated by NIH program staff based on scientific and technical merit of the proposed project, and will include things like the relevance of the project to the HIVE and HuBMAP, the expertise, commitment, and track record of the project team, whether the vision, benchmarks, deliverables, timeline, and budget are congruent with the tasks of that component, and whether the proposed work will realistically deliver the required capabilities as part of an actively managed other transaction award. Teams which then submit a letter of intent by February 28th will be notified by March 9th if they're invited to submit a key application. So the key here is that these applications are invite-only based on the LOIs. And collaborative applicants should also be aware that only a subset of proposed components may be invited to submit that full application. In May, a subset of OT applicants will be invited to present their applications and address questions in an interactive review panel, and this will take place in June. This will be a video or in-person interview to clarify any questions about the application, and please note that the NIH will not be paying for travel for those in-person interviews. And as far as evaluation of applications, they'll be evaluated by an appropriate review group convened by the Office of the Director of the NIH, and we'll be using the criteria described in Section 5.1 of the SOA, and this will be done by a group of NIH program staff and outside experts. An assignment to review group will be shown in the ERA Commons. And then around July or August, the NIH HuBMAP team will negotiate the budget, benchmarks, deliverables with selected applicants, and then make the award after successful negotiations. So LOIs can be submitted to the hubmap@mail.nih.gov email address by February 28th. And I encourage you to read the announcement for full instructions, but just an overview. There'll be just an overview of the project and the strategy, 2 pages, personnel qualifications, the estimated budget for the first year, and up to 3 letters of support as appropriate. And just please note, a separate LOI will be required for each component. As far as the application format, the SF424, please be sure to include your invitation to submit in the application. And as far as the research plan, just please note this is not a typical NIH grant application. So for the vision and overview section, don't consider this to be a traditional specific aims page for hypothesis-driven research. This section should provide an overview of the component and the vision for how that component will work as part of the HIVE and the HuBMAP consortium. Applicants should briefly state the goals of the component and how deliverables will address the aims and the goals of the SOA. And you should also provide a summary of any major tasks to be accomplished with milestones, benchmarks, a timeline, and deliverables. For the scientific, technical, and management strategy, again, don't consider this to be a traditional research strategy. The objective of the FOA is to identify projects and teams who can develop the data infrastructure and collaborative community needed by HuBMAP and the broader research community, as described earlier in this webinar. So this section should be structured to provide sufficient information to measure the applicant's likelihood of producing the deliverables and achieving their objectives. The next step is our HuBMAP OT objective review process. Again, this is not a typical NIH peer review. Objective review interviews will have a selected group of applications invited to participate again in that interactive review panel in June for clarification of any questions that come up from reviewers. This will be a 1-hour interview, either in person or by videocast. Again, NIH is not supporting travel for these interviews. And then scored applications will receive just a brief summary of review discussion. For objective review criteria, I encourage you to read the FOA, but this will be different for our HuBMAP FOA. We're going to use weighted scores. You can see them here. This is merit of scientific, technical, and management strategy, personnel qualifications, and institutional commitment. And with that, I'll turn it over to Terry.
Hi, I'm Terry Jurasic with the National Institute of Mental Health. I will be the agreements officer for these. And as we have said many, many times, these are not grants or cooperative agreements or contracts. They allow for much more flexibility, and that is true with the way they are awarded as well. Award segments are milestone-driven and can range from 4 to 12 months. However, no out-year commitments are included at that time. Future support is contingent on several factors, and NIH may extend or terminate for convenience. Any extensions will be fully negotiated. So these are handled very differently than your traditional NIH grant. This is a partnership, and NIH is invested in the success of the program. There are some similarities to NIH grants for submission and management, such as ERA Commons registration, SAM, cost principles, record management, payments, and many public policies apply to OTs. On here, we mentioned, as an example, DUNS. I just want to mention that right now, DUNS is required at submission, but we're working towards getting it required at the time of award instead, due to the different types of applicants we may have. I believe that could be happening almost any day. Conversely, there are several differences, such as the FAR and the NIH policy does not apply. HuBMAP has its own policy guide. The format and review of the applications, as Jessica just mentioned, could be very different. There's a letter of intent is required. You must be invited to submit an application. Review criteria is different, and you must have an interview, among other things. The funding segment length, no future commitments, and it can be terminated or extended by an NIH. NIH can also propose changes. So again, this is very much a partnership, and pretty much everything is up for negotiation. And I think it's back to you.
Yeah, so this is just a list of some of the web resources that we have created to go along with both the HuBMAP program. The ones listed at the top, if you have general questions, you can send them to our general mailbox, which is hubmap@mail.nih.gov. All these materials are available on our website, which is commonfund.nih.gov/hubmap. We also have a mailing list where we send out details of both activities within the HuBMAP program as well as related activities across NIH and some outside groups as well. And specifically to the other transactions that we have talked about today, we'll post these slides after the webinar. There's also a lot of appendix material that we haven't covered in this presentation so far, but which you can look at offline in your own time. And again, if you have questions related to that, please contact us by email. But we wanted to make sure that we had enough time during this 1-hour presentation that to allow time for questions. So if you'd like to submit any questions, please use the Q&A box in WebEx, And just again to reiterate that these are not grants, and we strongly encourage you talking with us, particularly if you're thinking about submitting a letter of intent, and also to engage your sponsored research office or your authorized representative in this process, because again, from previous experience with other transactions, I think it can be a shock to some of them about how these awards are managed. So I think it's important to engage those staff early in the process to make sure they're aware of what the implications are if you're submitting an application in terms of how these awards are managed. So again, finally, just remember that an approved letter of intent is required before an application can be submitted, and those letters of intent are due February 28th. So far we have no questions, or at least we've addressed all the questions that have been raised so far. Yeah. So in terms of non-traditional partners, yes, it includes both commercial entities, so large and small. So we're open to different kinds of partnerships. And again, if you look in the details of the RFA, in terms of how we, for example, describe cost sharing, it's not required. We're certainly open to negotiation, particularly if you are bringing in multiple partners who are interested in sharing costs or contributions in kind towards a project. We had a question about how many for each mechanism are expected to be funded. I think for each of the 4 components, I think the answer is a little bit different. I think we're expecting primarily to make 1 award per component. I think very much in the case of the infrastructure, that's true. I think we're only expecting to establish 1 platform for doing all of this work. I think where there may be an exception, for example, would be on the tools component, particularly if there are a number of strong proposals in that area. What we may do is we may combine them into a single award. We may integrate them all together. I think that's our strong preference, is that for each of those 4 components we have a single award. But I think depending on the nature of the applications and how they're reviewed, we're open to other ways. We had a question about how do you decide on the amount of an application. I think John, that's your question in terms of how much money do you ask for. I think the guide there is, if you look at the tissue mapping center and the way the budget for the tissue mapping centers are proposed to escalate over the first 4 years of the program, I think you can use that as a guide for thinking about the components of the HIVE. And in particular, I think we're not expecting an equal distribution of money across the 4 components. For example, we would expect that a lot more funding would go into establishing the infrastructure in the early years of the program, whereas I think as more data becomes available for the mapping, I think the mapping component, we would expect the budget for that to grow later in the program. So I think the numbers that are listed, I think we only list numbers for the first year. I think I would use that as a very rough guide, and then think about the escalation of the budget in terms of what we've outlined for the tissue mapping centers. So Nathan had asked a very sort of similar question along the total budget. So $2 million is the total budget available for the first year for all the components. So I wouldn't look at it as an equal division between all the components, but I would look at it as, I think each component will evolve separately, and again, that's why we're funding them as 4 separate awards. What are examples, David asked, what are examples of biological projects that fit the intent? So the focus of the program very much is on normal human tissues. So all of the data that we would expect to go into the HIVE would be of human origin, and it would be from normal, or at least non-obviously diseased tissue. So that's really the scope of the the type of data, I think we can then parse it out in terms of the different assays, and if people are interested in that, we can talk a little bit more about the data types that we would expect the HIVE to accommodate. So Kitty had asked what types of commitments are allowed in terms of institutional commitments, and the fact that 10% of the 10% of the weighting for the objective review process is assigned to institutional commitments. I think there we're very open to discussion about what institutional commitments people are willing to make. There's no real limitations there. I think certainly we're interested in leveraging the establishment of these components against existing resources that exist within institutions. Others, we're also looking for institutional support, as for example, you might see for any other type of NIH center. Do you expect to have human, so Carl had asked, do you expect to have human subject data? Yes, so I think the expectation is that the HIVE will likely have to deal with human subject data. That we can't give you a precise answer to this until we establish the tissue mapping centers. But given that at least some of the proposals we're expecting from the tissue mapping centers RFA will involve human subject data from living subjects, then I think the handoff of that data to the HIVE would include human subject information. So I think that any proposals coming in terms of particularly for the infrastructure component, should be prepared to handle identifiable human subject information. So Carl had also asked, is it possible to get an estimate of data types and volumes to help guide costs? We have talked extensively about this, and we could put our finger in the air and guess about the extent of the data. I think it's easier to talk about the data types, and I think what we're expecting is that we would have a significant volume of imaging data. So we're clearly looking for the HIVE to be able to ingest that imaging data, be able to work with the metadata associated with that, and to be able to archive it in a way that's cost effective as well as can be curated well. And alongside that, and we're looking for integrated solutions here, will be the omics data, and again, that can cover a wide range of different data types from, for example, RNA sequencing data through to proteomics data. So we're really looking for a flexible, scalable, and as Ajay mentioned, cloud-first approach to the data types, so that we can try to integrate them all together in one entity, which is going to be the HIVE. In terms of volumes, if we had to pick a number, and this is where all my colleagues will give me a funny look, I think the expectation is, we're easily up into the petabytes range for the data generation during this program. I think it's fair to say we don't expect to see a lot of data generation going into the HIVE for the first couple of years. So there will be a runway into this process for the HIVE to get set up. The first year of the program will be the setup phase. So a lot of the discussions will be around trying to smooth out that interface between the tissue mapping centers and the HIVE, to agree on the formats and the standards that we're using. So I think the first year will be a lot of that, and the second year will then be a lot of pilot studies to look at what happens when we try executing this in practice, doing cross-site comparisons of different, the same assay being done at different sites. So I think the expectation is the first year is really about establishing how all the different components of the program will work together. The second year will be much more focused on trying to get some of these pilot projects up and running and making sure that the pipelines operate smoothly. And I think then towards the end of the second year is when we expect to see production quality or publication quality data being submitted into the HIVE. So we had a question from Chris. So the FOA mentions harmonization and adopting existing metadata standards. Is targeted development of these standards in scope of the FOA? And is this a collaborative component or infrastructure component? So I think that the question is, yes, so specifically to answer the question, yes, it is potentially within scope. We're interested in using existing standards where possible. You know, we recognize that those standards don't cover all of what we're interested in doing. So development of the standards, I think, is definitely part of what the HIVE needs to be thinking about. And that's really part of the collaborative component of the HIVE overall. You know, the collaborative component is both this inwardly focused, making sure the consortium operates well as externally focused, and that's really engagement with external partners. So that, you know, again, that really brings in this standards part. We had a question, is there any difference between HuBMAP and the Human Cell Atlas project? So we cover this in much more detail in our frequently asked questions. You know, I think the quick answer to this is the HuBMAP program is very focused on understanding the organization of tissues and how they relate to the overall function of those tissues. The Human Cell Atlas is a grassroots international effort that has a much broader scope of interest and covers a much wider range. So I think the way to look at it is HuBMAP's a very targeted program in terms of thinking about the spatial organization of tissue. It's going to run for a limited length of time. It's got defined goals. Those are obviously to some extent complementary to what the Human Cell Atlas project is doing, but you know, I think the best way to look at it is they're two quite different efforts and programs, but obviously we're interested in working together. So we had a question, should the HIVE be similar to a library with experts curating content, or more like a marketplace that facilitates easy code exchange and combination by many? So, you know, the quick answer is the purpose of the HIVE is really to act as the central repository for the program initially. So it's very much, I would say, more on somewhat on the curated end rather than a marketplace. Certainly the expectation is as the program moves forward, and this is more towards the end of the scale-up phase and into the production phase, we are very interested in partnering with some of the similar atlasing efforts at NIH. You know, I'll pick the BRAIN initiative as an example. They have their cell census network, and we're interested in working closely with them to make sure that our data collection, as well as the way in which we're archiving the data, is compatible with the data they're collecting. So at some point, we're not quite a marketplace approach, but we're interested in certainly being able to integrate what HuBMAP is doing with a lot of these other atlasing efforts.
So if I may, I think it's probably a combination. It's more like Wikipedia, if you could think of that, where people participate, collaborate, but they also check on each other and curate the data, right?
Yes, yeah. And you know, we also describe in the RFA that the plans are that we'll establish some kind of associate membership type situation where individual investigators will also be able to submit data and make use of the HIVE facilities. Again, this is really set up for later in the program during the production phase. But in terms of thinking about where the HIVE is going, you know, it's important to recognize that this will not just be a data platform for the program, but eventually we hope to broaden it out beyond that. So we had a question about, do we have a sense of the ratio between new tool development by the HIVE versus integration of tools developed by the sites generating data? So this is a really great question. You know, so I'll parse it out even a little bit further than that. So for the tools component of the HIVE, I think really what we're interested in there is a suite of tools. So we're really looking for a combination of both existing tools that maybe need to be developed further, as well as the development of new tools. So I think we're very much looking for a suite there, covering search, browsing of data, as well as visualization. Those are the three main topics, but there's also more detail given in the RFA. So I think that's really what we're looking for in terms of the tools component. So we're not interested in one particular tool or technique to be developed, but really a suite of tools. And each of the tissue mapping centers also has a data analysis core, and within each of those tissue mapping centers, there will also be some analytical tools developed. I think the way to look at it is those tissue mapping centers are very much focused on the tissues they'll be analyzing, whereas what the HIVE should be focused on is more of a body-wide approach. So so it's very much the integration of all the data coming from each of the tissue mapping centers, integration with existing data, integration with emerging programs that are also doing atlasing efforts. So I think there will be some complementary nature in terms of the tools, and I think that's where the collaboration will come for some of the tools development, but also the tools needed for the HIVE will also be different given the nature of the types of integration they'll be doing. Any? No, we've got one more question. In addition to analytical tools, would you like to integrate computational models as well? Yes, very much is the quick answer to that. You can see in the RFA, we really try to outline that we're interested in making sure that the data that we're generating can be then included into computational models. And again, I think this is where we're interested to see what people propose as part of their applications. What are the best environments in which to do both. I'll go back to the tools component and mention the visualization, searching and identification of the data. What are the environments to do that in? Can those be easily integrated then with some of these computational models? Is it best to do those as separate approaches? So I think computational models very much part of the program, and we're very interested, and certainly later in the program that will be a component that comes in more strongly. Would any of my NIH colleagues like to add anything that we haven't?
The only thing I would like to add, I'm Rebecca Claycamp. I'm working in support of the agreements officer. If you're not from a university and you don't have a lot of experience working with NIH, I might recommend early to have your business office contact us so that we can help you kind of see what you're getting into, so that there aren't any unpleasant surprises as you plan your award, because the end product and how we are going to negotiate this with you will have an impact at some level on how you want to design it. So do reach out to us.
Great. So if there are no other questions at this stage, we'll post the slides deck that we presented today, along with the additional information. I think altogether we have about 35 slides. I think we only talked about 23 of them. So there's a lot of additional information. We'll post that after this webinar. It'll take us a couple of days to post the audio from this webinar, but we'll also post that on the website. The frequently asked questions are already up there. The policy guide is there, details of the public policies as well. So there's a lot of existing information available on the website. Please ask us questions. And just finally, to reiterate the deadlines for the announcement. So As I mentioned, we strongly encourage you to talk with us, preferably sooner rather than later if you are planning on submitting a letter of intent. And again, not just investigators, but also your sponsored research or business offices, bringing those people into the conversation early on. The letters of intent need to be submitted by close of business on February 28th to the HuBMAP email address, so that's hubmap@mail.nih.gov. And for those letters of intent, you must propose one component and only one component per letter of intent, but you can also submit a coordinated set of letters of intent if you're interested in applying for multiple components. And there's details in the RFA for exactly how to do that. Then within 2 weeks of February 28th, so essentially by mid-March, we'll get back to you and let you know whether you've been invited to submit a full application, and those full applications then are due a month later by April 16th, and those will be submitted through the normal ERA Commons approach. And again, if you have questions about that, we're more than happy to sit down and discuss that process if you're not familiar with it. Then, following that April 16th receipt date, we'll follow up with people who are going to be invited for interview. So that'll be sometime in mid-May. We'll notify the applications that have been identified as the most meritorious at that stage to come either in person for interview, or we'll set it up via video cast, and those interviews will be in June. And then be prepared for a negotiation phase once we have decided the applications we're interested in funding, and that negotiation phase will be in July and August. So in terms of thinking about a potential start date, the start date for these awards would be sometime in September. So thank you again for calling in for this webinar. Again, we strongly encourage you to talk with us if you're interested in submitting an application, and certainly the earlier the better, given some of the differences about both the way this RFA is structured as well as the mechanism that we're using to fund these awards. Great, so if there's no final questions, thanks again, and we'll post this material online later today.
Bye.